So, let me show you what we're finding. These are veins. Now, what we do with a bone is we put it in a weak acid. Remember when you were a kid, maybe you took a chicken bone and you put it in a jar of vinegar and left it for a week and you brought it out, it was all floppy. That's decalcification. The vinegar is a weak acid, so we use a weak acid, we soak the bone in it, and then all the tissue structure that's in the bone falls out on the Petri dish. And that's what you're seeing here. These are all veins that were up in the 3D bone, right? And they dissolved out, and now they're laying on the bottom of the Petri dish. So we find veins, and we find vessels. But we also find vein valves. What's a vein valve? Well, when your heart pumps, it's pushing blood through the arteries, right? The blood vessels. And then when the blood returns to the heart, it comes back through the venous system, the veins. Well, there's little valves in there. Why? Because the blood pressure is lower in the veins. So when it pushes blood up, the valves open, the blood gets pushed up, and then they close. They're holding that blood in place. And so it ratchets it up. Each time they open and close, open and close. We have found the actual vein valves from the veins. Here, I don't know if you can see it, they're stained kind of pink. I stained these vein valves with an RNA locating stain. That pink color, that's all RNA. You've heard of DNA, RNA, deoxyribonucleic acid. It's a single strand. This is all single strand DNA that's returning a signal. You're going to see more of that later. But if you look at this picture, you see the ring of the valve, and look at these little vellums of tissue. There's actually, it's a bicuspid like your heart. One goes up, one goes down. So that's how they open and close. Now I want to show you a video. I'm going to show you a video of a vein valve that I found on the bottom of a Petri dish. If you look really quickly, you'll see those little things swimming around in there. See those little dots in there? Those are bacteria that were in the bone, soil bacteria, that came back to life because of the water. They make spores. Bacteria go into spore stage. And so they're swimming around inside of this vein valve, and they got stained with the RNA stain also. So they're glowing red, just like the vein valve is. See them glowing red? So that means they couldn't get out of the vein valve. It's actually closed upon them. So it's a fully functional, intact valve. Do you understand my meaning here? This is a fully functional valve that's supposedly 68 million years old. This little piece of tissue is 1/100th the diameter of a human hair. How can that still be here? That should have decayed to nothingness. Apoptosis, programmed cell death, should have rendered that to dust in a matter of years or even decades, right, at the outside. So there's no way, this is impossible, that we're seeing these things. In fact, I was actually able to use a regular tissue stain, kind of like food coloring. And this tissue acted like a normal tissue, and it took up the stain. Look at how dark this is. That's a venule valve with a stained cuspid in it. So it behaves like regular tissue. Shouldn't be, but it is. What else are we finding? We're finding nerves, ladies and gentlemen. This is a nerve from a condyle, which is a softball-shaped bone at the base of the Triceratops skull. It's called a condyle. And it was full of nerves. And I've found nerves now in just about every bone we've looked at. Now, nerves are collagen with axons. There's neural axons in there. And they're wrapped in myelin. They're wrapped in fat. And you can actually see the wrapping. See the wrappings? And I'm going to show you this in more detail because I thin section them. This is what we do in biology. In order to understand the structure, we thin section things to see the structure. Here's a little higher magnification. And the neurosurgeon who did my neurosurgery, I sent him the paper to review it first, and he was astounded. This is a guy who knows nerves. He operates on them all day long, every day. And so he recognized these little white bands in here. These are called the bands of Fontana. You can see them faintly all the way across. Even down here you can see some of them. That's a dead ringer for nerve. If you have bands of Fontana, you've proven that you have nerve. So we're finding these structures. And what I did is I took a chicken bone and I dissolved it in acid and I got a nerve out of it. That's a chicken nerve. Compare it to the condyle nerve and to the vertebral nerve. They look almost identical, don't they? The coloration's a little different. That's an artifact of the polarized light microscopy. But structurally, these things are identical. But it gets better. When I thin sectioned the chicken nerve, I took off the top layer of the nerve. You can see the myelin sheath. See that fatty covering? So that's what wraps the outside. But inside of that is a cross hatch, almost, what would you call this, a tartan weave or something? I'm not a textile person. But it's a weave. See the cross? It's going this way and it's going this way. That's in the chicken nerve. Look at what I found in a decalcified triceratops horn slide. This was horn. This is the same wrapping. It's just come undone. Do you see that? And here it is in polarized light, so you can see the same kind of colors. See that? So we are circling around, I think, one of the most important finds in paleontology. What I also did is I thin sectioned the nerve further. So here's the chicken nerve when I first started sectioning it, and then I cut all the way through it. And you can see all this structure in here. But the other thing you could see is these band markings that pretty much go all the way through. So I know I have nerves. There's no question about it.